Good day, everyone. So for today, we will discuss the last topic, which is the bacterial species of limited pathogenic significance. So these are a heterogeneous group of gram-negative bacteria that contains organisms occasionally producing disease in domestic animals and also a number of bacterial species of uncertain pathogenicity commonly isolated from clinical specimen. So we'll start with Acinetobacter species. These are gram-negative Coccobacilli organisms, easily isolated on standard culture media, are environmental organisms which cause opportunistic infections in humans and animals, especially in hospital settings. And they are the most commonly isolated are species isolated are Acinetobacter baumannii, Calcoaceticus, and Wolffi. Acinetobacter have been isolated from different clinical conditions, including post-operative wound infections and septicemia. Then we have the family Bartonellaceae. The members of the family Bartonellaceae are small, thin, short, and slightly curved gram-negative rods. Until the early 1990s, the genus Bartonella consisted of only one species, which is Bartonella bacilliformis. Genus now contains all the species that were once included in the genera Bartonella, Rockalimea, and Grahamelia. Genus Bartonella has been placed into the family Bartonellaceae, which has also been removed from the order Rickettsiales. So the most important species of Bartonella that is cause of cat scratch disease in humans is Bartonella henselae, formerly classified in the order Rickettsiales and contains 19 recognized species. Gram-negative, slightly curved rod grow on blood-enriched media, require 3 to 4 weeks to develop. Organism is carried by healthy cats, transmitted by cat flea to other cats. Mode of transmission is by arthropod vector, while cats has no clinical signs. Humans, cat scratch disease that is seen as a small skin lesion which progresses to an ulcer and then heals. Lymphadenitis, fever, malaise, and headaches are seen, causes bacillary angiomatosis in immunocompromised individuals. Transmission in animals is via vector, which are the cat flea, Tenocephalides felis. While in humans, transmission is via the direct contact from cats through scratch, bite, or by cat flea bite. So this is how it is being transmitted in humans from animals. That's why the disease is called as cat scratch disease, which occur as a small skin lesion that may later ulcerate and then heal. In 1 to 3 weeks, usually there is inflammation of the lymph nodes, especially in the axillary area. So it may resolution can be seen in weeks to months. Dissemination can be seen in immunocompromised hosts. So this table shows the bacterial species with limited veterinary significance. So we have mentioned the Bartonella henselae. In humans, the disease is cat scratch disease. In cats, there is no clinical signs manifested. Next, we have Chromobacterium violaceum in pigs, dogs, and sheep that is known to cause opportunistic infections. It is also known to be a saprophyte in soil and water. Flexispira rapini in sheep is cause of sporadic abortion. And Helicobacter species, which we have discussed already, included in the Campylobacter species, like the pylori that is cause of gastric and duodenal ulcer. We have in ferrets, we have the mustelae of Helicobacters affecting ferrets, dogs, cats, resulting to chronic gastritis and gastric ulceration. It is found in gastric mucosa and intestines. Next, we have the megabacteria, which affects barge rigors, resulting to megabacteriosis present in the proventriculus of birds. Ornithobacterium rhinotrachale is affecting chickens and turkeys, resulting to respiratory infection or disease. Rhimerella anatipestifer, which was formerly named as Pastorella anatipestifer, affecting ducklings, causing septicemia or the new duck disease. Streptobacillus moniliformis in Turkey is the cause of septicemia and is a normal inhabitant of the upper respiratory tract of rodents. Chromobacterium violaceum is a gram-negative rod, motile and facultative and aerobe, grows on MCA and nutrient agar with production of violet pigment, catalase and oxidase positive, found in soil and water of tropical and subtropical regions. Barbariship is associated, for barbariship, the organism is associated with acute pleuropneumonia. In humans, pigs, and dogs, septicemic infections have been reported. Pigs, dogs, and sheep may cause opportunistic infections. This is the appearance of the violet pigment of the Chromobacterium violaceum. This is a chromogenic bacteria. Next, we have Flexispira rapini, provisional name for a curved motile microaerophilic gram-negative bacterium, constituent member of the flora of the intestines of laboratory mice. In sheep, reported sporadic abortion in USA and UK. Aborted lambs have multifocal hepatic necrosis resembling hepatic lesions caused by Campylobacter species, especially subspecies fetus. Campylobacter fetus, subspecies fetus. Megabacteria is a provisional name for a group of large gram-positive rods measuring 20 to 50 by 3 micrometer. They are found in the superficial mucosal glands of the lower portion of the proventriculus of budgerigars. Catalase and oxidase negative, facultative and aerobes. pH in the proventriculus changes from pH 2 to pH 7 to 8. Organism grows optimally in blood agar, producing small hemolytic colonies for 2 days with 10% CO2. So in budgerigars, the organism is present in proventriculus, implicated in megabacteriosis, which is a wasting disease among this bird species. Clinical signs are weight loss, diarrhea, and vomiting. Treatment is amphotericin B. Next, we have Ornithobacterium rhinotrachale, pleomorphic gram-negative rod, grows on blood agar, produce small grain non-hemolytic colonies. Aerobic capnophilic with 5 to 10% CO2, enhance its growth. Oxidase positive and catalase negative, causing respiratory disease in chicken and turkey. Treatment is using amoxicillin or giving amoxicillin in drinking water, 200 ppm, parts per million, on 2 consecutive days. Next, we have Rhimerella anatipestifer, non-motile as a catalytic gram-negative rod, grows optimally on enriched media in an atmosphere of 5 to 10% CO2, non-hemolytic on BAP, no growth on MCA, affects duckling up to 6 weeks of age. Older waterfowl, turkey poults, chickens, and pheasants are also affected. And in ducks, there is septicemia. So the disease in ducks is precipitated by stress. Clinical signs are ocular and nasal discharges. There are neck and head tremors and incoordination seen. Mortality reached 70%. Postmortem findings are fibrinous pericarditis and peritonitis, meningitis, and fibrinous erysiculitis. Treatment and control, intramuscular injection of streptomycin or dihydrostreptomycin in the early stages of the disease. Sulfadimidine sodium can be given in drinking water for 3 days, and there is a bacterine and a live virulent vaccine available. Next, we have Streptobacillus moniliformis, a highly pleomorphic gram-negative rod, occasionally causes outbreaks of bronchopneumonia in laboratory rats and mice, and cervical lymphadenitis in guinea pig. Humans, there is Haverhill fever and rat bite fever. In Turkey, normal inhabitant of the upper respiratory tract of rodents, and the transmission in Turkey is septicemia following rat bites. So these are bacteria of uncertain, hindi pa established kung yung kanilang pathogenicity. They were commonly isolated from clinical specimen. We have Achromobacter, commonly found in the environment. Xylooxidans. Achromobacter xylooxidans is an emerging nosocomial pathogen in human. Occasional reports of opportunistic infection in animals has been reported. Alkaligenous species, like Alkaligenous fecalis, is a saprophyte. They are saprophyte occasionally isolated from the intestinal tract of vertebrate animals. Branhamelia species isolated from nasopharynx and conjunctiva of clinically normal animals. Flavobacterium species present in soil and water. Nosocomial infections in humans. Some species are pathogens of fish. And we have also Neisseria species. They are present in the nasopharynx and conjunctiva of many animal species. And that ends the topic on bacterial species of limited veterinary significance. Thank you and have a good day.